This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Okay, well, welcome everyone this afternoon to Gothami's confirmation seminar. Just a bit of an introduction about Gothami. She graduated from the University of Paradinya in Sri Lanka in 2013. And then she worked, she did an internship at the VRI in Sri Lanka, followed by another one at the National Zoological Gardens, also in Sri Lanka. And then she became a vet surgeon at the City Pet Hospital. And then from there, she joined us in 2019. And as we all know, it's been a pretty tumultuous few years with COVID, and Gothami had a baby in between. So she's definitely shown an incredible ability to persevere and take on all the challenges that have come up to her. So without further ado, I'd like to welcome Gothami and wish her well with her confirmation seminar today. Thanks, Gothami.
Thank you, Vern, and I welcome you all to this seminar. So the title of today's presentation is Identification of Host and Parasite Responses in Early Flystrike Lesion. The focus of the today's talk, Australian sheep blue fly, was introduced to the Australian fauna in 1880s with the introduction of Vermont merino sheep breed. Since then, it has become a major pest in the sheep industry, causing a significant economic impact. And it's been estimated that the cost of the Flystrike to the Australian sheep industry is around $324 million per each year. And let me briefly introduce the sheep blue fly. And cutaneous miasis, or the Flystrike, means the infestation of sheep by the parasitic larvae of Lucilia species. In Australia, more than 90% of the Flystrikes are initiated by the Lucilia caprina. Most of the cases occurs in the breech area. You can refer this picture. More than 80% of the cases occurs in the breech area, but it's not uncommon to see cases in the body or in the places like feet. Now let's briefly go through the life cycle of the Lucilia caprina. So it starts when gravid female flies go and oviposit on a susceptible sheep, and usually flies to lay about 200 eggs, and they usually lay eggs as a group so they can prevent the desiccation of the eggs. Within 8 to 24 hours later, the first instar larvae would come out from these eggs, and then they undergo 2 developmental stages within the sheep to become second star larvae and third instar larvae. This fully fed third instar.
Talking about the pathogenesis of the disease, as I mentioned before, female flies, they go and oviposit on the susceptible sheep, and within 8 to 24 hours later, first instar larvae would come out from these eggs, and they have these small spines in their mouth, which they use to abrade the skin. Same time, they will release a lot of enzymes which degrade the host tissue. And these mechanical and the chemical reaction would stimulate the inflammatory reaction from the host tissues, and the collective action of host immune response together with the mechanical and chemical damage of the larvae would result in a rapid development of an exudative wound. So initially we would observe behavioral changes like tail twitching and biting the affected area. Then we would observe physiological changes like fever, anorexia, and the reduction of body weight. And this would get complicated with the secondary bacterial infection, which results in ruminal stasis, sepsis, ammonia toxicity, and if not treated, like it would result in death. And we would also see a reduction in the performances, like reducing the wool cut and the quality, and sometimes lamb losses in the affected ewes. So now let's talk about the predisposing factors. Anything that would make the fleece of the sheep smelly and damp would result in Flystrike. This merino sheep breed, they are susceptible because they have a lot of wrinkles and dense wool cover in the body, and the wrinkles and the ability to fleece to retain the moisture would provide the favorable microenvironment for the larvae, so that will result in the Flystrike. Similarly, urine and fecal staining in the perineum, as you can see in the first picture, it would attract gravid flies. And bacterial and fungal infections in the skin, like fleas rot and dermatophytosis, would result in the strike. And fecal soiling of the fleas, especially in animals that have diarrhea, you can see in the second picture that the breech area is covered with jags. Those animals are highly susceptible to get the Flystrike. And presence of lesions like shearing cuts and mollusing wounds, and especially if the animals are already having Flystrike lesions, then they are susceptible to get Flystrikes. Not only that, but climatic conditions like high temperature and low wind speed would result in more fly activity, and therefore there's a high chance of having Flystrikes in sheep. And let's see what are the current treatment and control methods we have. So the treatment is basically centered around using insecticides. There are about 6 groups of insecticides that we use currently. And fly trapping is another method. We can use odor-baited catching devices to catch the flies and to reduce the fly population. And there are other sheep management practices like shearing and crutching, which can reduce the susceptibility of the sheep. Here what we do is, in shearing, we remove the fleas from the body, and in crutching, we remove the fleas in the breech area of the animal. Mollusing is another method that we use. Here what we do is, we remove a part of the skin in the breech, in the perineal region. And once this wound is healed, the area we get is devoid of wrinkles and the skin folds. So there's a less chance of having fecal contamination in the breech area. And it's highly effective in controlling the breech strike. Tail dogging is another method that practiced to control the Flystrike. Here we amputate the tail at the third caudal vertebra. So by doing this surgery, we can reduce the contamination of the tail and the breech area from the fecal matter. It's important to regularly inspect the flock so we can identify the animals with the early Flystrike lesion, and we can treat them. There are novel control strategies that are still under the experimental level. For an example, use of entomopathogens to control the Flystrike, like Bt toxins and fungi like metahesium. These methods can be used to control the Flystrike. And genetic manipulation of the fly using methods like CRISPR-Cas9 and RNAi can also be used to control the Flystrike, but all these methods are still under experimental level. And vaccination is another method that we can use to control the Flystrike. There have been a lot of vaccine antigens that have been studied, including natural antigens and concealed antigens. And some of these antigens showed retarded larval growth in vitros and in vivo assays. But there's no commercially available vaccine at the moment. Selective breeding of sheep is one of the methods that we can use to produce animals that are resistant to Flystrike. It's a long-term procedure. What we usually do here is that we select the animals based on certain indicator traits like urine color, wool color, and the presence of less jags in the breech area, presence of less wrinkles, and resistant to the fleas rot. So AWR has developed a scoring system, and we can use this scoring system as a guide to select our animals. And animals who have higher scores can be removed from the flock, while the animals have a good score like score 2 can be used in the breeding program to get a progeny that are less susceptible to the Flystrike. So in 2005, AWI has started funding a breeding program, which was conducted in 2 climatic zones in Australia. One was in Western Australia, and the other one was in Armidale, which belongs to the summer rainfall zone. What they have done here is that they have monitored the animal, and animals that are not struck by the blue flies, they were selected and used in their breeding program. So they have maintained this breeding program for about 15 generations. And once they analyze the data, what they have found is that the proportion of the total variation they have observed in this animal can be explained with the differences in their phenotypic characteristics. So some of these resistant animals have physical characteristics like less breech wrinkles and less gaps and less urine coloration in the breech area. However, a proportion of these resistant animals, their resistance could not be correlated with these phenotypic characters. So then we get the question, what other factors could explain this resistance? So assuming that it could be their immune response, so this resistant animal could be immunologically different to the non-selected animal. Assuming this fact, we use this research block in our study. So we got 7 Flystrike resistant and 7 Flystrike non-selected sheep from the CSIRO facility in Armidale, and we used those animals in our study. So we hypothesized that this resistant sheep, they have different cellular and cytokine profiles when we compare them to the non-selected sheep. And secondly, we hypothesized, in this second hypothesis, it doesn't have anything to do with the Flystrike resistance or non-selection. Here what we assumed is that the larval molecules produced at the early Flystrike lesions, they are essential for the survival of the larvae. So based on these 2 hypotheses, we designed our study. So the objective of this study was to compare the host and parasite responses in animals selected to Flystrike resistant and animals that are not selected to the Flystrike resistant. And as the second objective, we were planning to identify potential drug targets or vaccine targets using the RNAi technique. Using those molecules we identified in the early Flystrike lesion. So here I have listed the aims that I'm going to use to test these objectives. The first aim was to compare the composition of the cellular infiltrate in Flystrike lesion from both selected and non-selected sheep once they are challenged with the blowfly larvae. So the aims that I have already completed will be highlighted in red color. And the second aim was to compare the cytokine production in the skin of these sheep. The third aim would be to identify the protein candidates using previous proteomic and transcriptomic data to knock down these protein molecules using RNAi technique. And the fourth aim would be to identify proteins that induce lethal effects once we knock down these proteins using RNA. And in this slide, I'm going to talk about the rationale behind this thesis, why I'm trying to look for an alternative control strategy like finding a new drug target or an effective vaccine candidate. As I mentioned in my previous slides, there are several control methods available in controlling Flystrike, but still we are facing different challenges with the current control methods. For an example, development of resistance to the existing insecticides. So some of the blue-fly species have shown resistance to the insecticides that we are currently using. Not only that, but contamination of wool and the environment with the use of insecticides, and acute and chronic health issues with some of the insecticides like organ phosphates. And selective breeding I have mentioned that selective breeding is one of the effective way of controlling Flystrike, but still it takes a lot of time. And mulesing is also effective in controlling the breed strike, but it has animal welfare concerns. So because of these limitations, it's important to look for other alternative control strategies. So then we have designed our study. First we selected first we used 7 Flystrike selected sheep and 7 unselected sheep. First I collected serum samples from both sheep group. Then I selected 8 random sites from the back of these animals. Out of these 8 sites, 4 sites were implanted with the dental plugs containing blowfly eggs, and the remaining 4 sites were implanted with the mock plugs. Then we monitored the sites for the evidence of larval hatch. Once we saw the larvae, we started collecting samples from both infected and the mock sites every 3 hours for 4 times. And at the 4th time point, we again collected blood samples and skin biopsies were also collected. And these are the samples that I have collected. So I got larvae from the resistant and unselected sheep. Then we got skin washings. Here I use a BVS containing proteinase inhibitor to wash the skin of the infected and mock sites. And then we also collected skin biopsies from the infected and mock sites of the resistant and unselected sheep. Then also we got serum from before and after the larval challenge. And in this slide, I'm going to talk about the materials and methods that I have used to achieve my first aim. So the skin samples were first fixed in 4% paraformaldehyde, and then the paraffin sections were stained using HNA staining. And part of the skin samples were embedded in the ICT, and they were frozen in liquid nitrogen. Then these frozen sections were stained for different immune cell populations, and the stained cells were counted under high power field. So these are the results I got. In this first slide, we can see in the first photograph, we can see it's from a control site. And in the second picture, compared to the first picture, you can see that.
So that includes everything from writing to speaking.
So in the second picture, you can see a purple color area showing an infiltration of leukocytes into the area where the larvae were feeding, compared to the control sites. But when you have a closer look, what you can see is accumulation of neutrophils, and also we can see some eosinophils as well. And when we analyze the immune cell populations of infected and control sites of the resistant and unselected group, what we saw is that there's a significant influx of the CD4 and CD8 positive cells, as well as CD45R, gamma delta, T19 and CD1 positive cells in the infected sites compared to the mock sites. However, the analysis of these cell counts in between resistant and unselected sheep showed that there is no statistically significant difference. In this graph, you can see that in between resistant and unselected sheep, there's no significant difference. But you can see a difference in between the control and in the infected sites. Similar observations can be found in the other cell counts as well. But to see this, so to confirm these results, I have used a statistical test. Here I use linear mixed model with restriction maximum likelihood method. So in the second column, you see the mean cell count of the infected sites. In the third column, you can find the mean cell count of the mock site. And in fourth column, you see the effect of the infection. And in the last column, you can see the effect of the resistant phenotype. For an example, if we see the CD4 cell count, in infected site, the mean cell count of is around 33. And in mock site, it's around 6. So there's about 5-fold difference in between the in mean cell count in between the infected and mock site. And if we see what makes this difference, is it because of the larval feeding or because of the Flystrike resistant phenotype? When we analyze the data, what we can see is that the effect of infection or the effect of larval feeding is around 0.001. So it's less than the p-value, which means it's statistically significant. And when we see the effect of Flystrike resistance on the differences that we have observed in the infected and mock sites, we see that it's higher than the 0.05, meaning that it's not statistically significant. So similarly, in other cell types, we can see that the effect of infection is statistically significant, but the effect of resistant phenotype is not statistically significant. So in conclusion, we can say that the presence of larvae resulted in large cellular infiltrate of leukocytes into the infected site compared to the mock site. However, the effect of Flystrike selection did not have a statistically significant effect on the differences we observed in the infected cell count, infected site compared to the mock site. So what does this mean? Either there was an inadequate exposure of larvae to generate a specific immune response, or the variation reported in this resistance is not due to the differences in cellular component of the immune response. So in my second aim was to compare the cytokine production in this selected sheep. There have been previous studies done to show that there's an upregulation of inflammatory cytokines as part of the immune response to Neisseria larvae. And in this study, I'm going to compare the cytokine profiles from both Flystrike selected and non-selected sheep. So here I have already extracted protein from the skin samples using this kit, and then I have to measure the protein concentration. Then I'm trying to use Millipex cytokine, Millipex O1 cytokine kit to detect the cytokines present in the skin samples. So I have done my first batch and got higher concentrations of cytokines in the standards and controls than the expected values. So I have to do a bit of optimization, and after doing some troubleshooting, I have run my another batch. So the subsequent one gave a satisfactory result. Now I'm planning to continue analyzing my study samples. And the third aim would be to use RNAi to identify the potential function of certain selected proteins on the larval survival. And here, so we use data from previous transcriptomic study, which was done by Dr. Perry and Prof. Bowles. So what they have done is they have allowed larvae to develop in sheep skin and also in liver samples and agar plate. And then they have analyzed or compared the transcriptome of these larvae. So what they have found is that certain genes are highly expressed when the larvae were feeding on the sheep skin compared to the liver and the agar sample. And they have identified these, they have selected these proteins to be used as candidate proteins for RNAi technique. And in the study that we have done using Flystrike resistant and non-selected sheep, we got our larvae, and from this larvae, we were trying to compare the ES products from the larvae that were raised in the agar plate. So we were comparing the proteins released by the larvae that were feeding on the early Flystrike lesion to that of the larvae that were feeding on a that were allowed to develop on a egg uptake. And once we analyze the proteins, what we have found is that certain proteins are highly expressed, or certain proteins are detected in high concentrations in the ES products of the larvae that were feeding on the sheepskin. So using the data of both of these studies, we got a list of candidate proteins, which I'm going to use later to do the RNAi technique. This is the strategy for RNAi technique. First, I have to identify the candidate proteins based on previous transcriptome and the proteomic data. Then I have to develop functional double-stranded RNA, and then I'm going to then I have to deliver those double-stranded RNA to the insect. So here I'm going to use embryo microinjection method because because it seems not effective compared to the other methods. And then I can assess the knocking down effect just by looking at the biological phenotype, or I can use qPCR to monitor the effectivity of the target gene knockdown. And I have already selected 7 proteins from the list that we have prepared using previous transcriptomic and proteomic data. These are the criteria that I have used to select my protein, whether the protein is predicted to be secreted during the natural infection, whether it's highly expressed during Flystrike, whether it has a lethal effect or not, based on the studies done on Drosophila using these proteins. And also I have looked at the potential function of these proteins. And these are the materials and methods that I'm going to use. So I use a fly colony maintained by Dr. Perry at his insectary in Bio21. First, I have to collect eggs in a piece of diced meat. Usually usually fresh eggs, which are less than half an hour old, are used to inject it. So these eggs are first lined up on the edge of a cover slip, and then microinjections of the RNA using RNA is done using anterior injection methods. And then these injected eggs are incubated at room temperature in a humid petri dish. Then we can monitor the larvae for the survival and the presence of desired phenotype. In this slide, I'm going to share information about a study done on a protein called mesh protein. It's an extracellular protein involved in smooth septate junction assembly. There have been studies done on this protein in Drosophila, and this study showed that the larvae that have this protein knocked down show death when they are at first instar larval stage. So Dr. Perry carried out similar study in his lab using the mesh protein, and what he has observed is that in this first picture shows the larvae of control group, so they are feeding happily on the nucleus, and in the second picture shows the larvae that has mutant that has the mesh protein knocked down, and these larvae instead of feeding, they are crawling away from the nucleus. So I am trying to use this same protein to get trained with the microinjection procedure and to decide which concentration of the double-stranded RNA can be used to inject to get an effective knocking down process. And these are the summary of results and progress to date. So I have found that there's no apparent difference in the B and C T cell population present in the wound initiation in Flystrike selected and unselected sheep. And also methodology for cytokine analysis of the skin samples have been established, and microinjections of the blue files have commenced. And in the coming weeks, I'm trying to select the genes for evaluation of those genes using RNAi technique. And this is my previous outline. In the first chapter, I'll be describing literature review, which includes the introduction to the fly and current control methods, and the rationale behind this thesis and the key objectives. In the second chapter, I'll be describing the materials and methods. And in subsequent chapters, I'll be describing the aims that I use to achieve my objectives. This is my time frame. So the black color shows the task that I have already completed. And here there's a gap, as Vern mentioned before, that I stayed in my home for about 2.5 years because of the COVID pandemic and some family commitments. So then in April in this year, I restarted my work. So there's about 2.5 year gap here. And the gray color, dark gray color shows the tasks that are in progress, and light gray color shows the tasks that I'll be doing in the future. And finally, I would like to thank all of my supervisors, including Prof. Bowles, Dr. Perry, Dr. Amstead, Dr. Baxter, and Prof. Shearling, and my advisory committee chair, Professor Carol Hartley, and Jane Smith from the CSIRO, Professor Graham Helfworth for supporting me with the statistical analysis, and Professor Prof. and her group, and Hubtamu for supporting me analyzing the immunohistochemical studies, and Dr. Ravegam and Tina and Shilpa, and all of my friends from the Building 400 and Bio21 and my family. And last but not least, University of Melbourne, Bio21 and Australian World Innovation for the financial support and for giving me this golden opportunity to study in this prestigious university. And thank you all for attending this seminar, and I really appreciate it. Thank you.
Thanks very much, Gothami. That was a good presentation. I'd like to open up the floor now to anyone who wants to ask any questions. I can't see any in the chat, but does anyone want to just pipe up and ask a question of Gothami? If I could invite someone.
I'll ask a question.
Okay, thanks, Carol.
How you going? Well done, Gothami. You did very well to get through the hiccups at the start, but we all heard you loud and clearly, so well done. It's a trick. So my question is about, so your initial experiments where you looked at the cellular infiltration in your animal studies. And I guess my question is about, I know why you only did one time point. That's quite clear because they're like sheep studies. So I guess my question is really about, has anyone ever done anything sort of ex vivo with sort of skin taken from sheep and then sort of in the lab, you know, because that would give you an opportunity to look at different time points rather than looking at, for the cellular infiltration stuff at least, sort of focusing on one time point? Because I imagine, but I don't know, I imagine like the differences between the resistant and the non-selected sheep are probably quite subtle in some of those areas. There's not going to be something that goes sort of blam, it's, you know, one thing. There might be a couple of different things. So I wondered if there was a way of looking at time points for different things like that.
Yeah, so previous studies done on sheep on the cellular infiltration, like within 48 after the infection. But this one, it's very, so we wanted to focus on the early phase of the flies, right? That's why we took our, that's why we terminate our study within 36 hours post-implantation of the eggs. Yeah, but I have no idea whether anyone has done it like using sheep skin samples on the lab.
Yeah, I don't know. I imagine that's a thing. I mean, I think, you know, they might be able to go anywhere, but yes, I understand why you've gone the earlier samples, but they might be to look at the early stages. But I just wondered if there's an opportunity somehow to look at sort of different time points. And I guess you can do that with your washers and things. You've got a few different time points with the washers earlier.
Skin washers. Yeah. So we looked at the protein concentration of those skin washes.
Yeah.
Yeah.
All righty, thank you.
Yeah, and just on that, Carol, well, if you isolate the sheepskin, you know, you lose the dynamics, because once you cut it out, it starts to die. So you could look at protein secretions from the larvae as they're feeding early on, but we can do that on the sheep. It's just to get the dynamics. What we wanted to do was do biopsies at time points across the line as the sheep were infected. But animal ethics killed it.
Fair enough. And the other thing, I guess, in that is neutrophils. Did you look at things like non adaptive cells like CD4 and CD8 you've got, and gamma deltas? I guess they're innate as well, but other things like neutrophils and
I tried counting neutrophils, but there's this severe infiltration, so it's really difficult to count cells by.
Yeah, are there markers? Are there neutrophil markers in sheep?
Not that I'm aware of. Not that I'm aware of.
All
right.
Specifically for neutrophils, I think there's.
JP, can you answer that?
Yes, not that I know of. The only thing that you can look at is through histology. You can do staining for the neutrophils, just a classical neutrophil staining, but it's not an antibody.
Yeah. Okay.
It makes it a bit more difficult.
Yeah.
In terms of looking at the time points, one of the things you could do is to have infections or put larvae on different parts of the sheep at different time points, and then kill the sheep and collect all the samples of 4 or 5 different points. So you could start in the morning and do, put them on, and then go along the whole day, and then at the end kill them, so that you have the same endpoint, but the time that the larvae have been on the sheep differs, because you have put different parts at different time points throughout the trial.
Okay, thank
So
you.
you could do that if you wanted to use one sheep and get all of the time points, at the end.
Yeah, like a countdown rather than a count up, which is what.
Countdown, that's exactly right.
Yeah.
Good on you, Gothami.
Yes, so there's a question from Paula about would animaletic dislike that cause of cumulative burden?
Yes,
The animaletics, why do they refuse to get you to take different samples?
it's because of the cumulative burden. Yes, that's why they asked us to terminate it.
So yes, Paula.
Have we got any other questions of Gothami? Oh, Simon Baxter. Simon, have you got a question?
Hi, Gothami. Yeah, I've got a question.
Hi,
Gothami, you're using RNAi to try and knock down gene expression. You're using microinjections. But I suppose the other way to deliver RNAi is through feeding experiments. For example, putting larvae on meat that has RNAi solution already added. So I guess, what are the advantages of microinjecting the RNAi compared to the feeding experiments? Can you explain why you're doing injections rather than feeding?
Yeah, so I read several papers, and what they have mentioned is that when you feed the RNAi, like when you feed the double-stranded RNA, like with the feeding with food or something, we don't know what concentration of double-stranded RNA go into the insect compared to the injection method. Because in injection method, we know what concentration we use to inject them. But in the feeding method, we don't know how much they will ingest and how much double-stranded RNA would go into the system compared to the microinjection method. So maybe that is one advantage we have over the feeding. Like we know what concentration we are injecting them. I suppose that answers the question.
Okay, so is your primary objective just to characterize perhaps the function of these genes, or are you looking for a potential to use RNAi as a potential drug for pest control?
My idea is to identify the function of the protein. So if I silence those proteins, if it would induce a lethal effect on the larvae, then I can target that protein to produce a drug target or a vaccine.
Okay, so how do you think you would target some specific proteins for pest control?
So it's like controlling the Lucilia species. Is that what you mean?
Yeah, I'm just wondering, like, these genes are responding to they're being overexpressed in larvae that are feeding on sheep. I'm just wondering, you know, how they could potentially be used for pest control.
Yeah, so let's assume if we target a protein that is crucial at the first instar stage. So if we can target that protein and if we can knock down that protein, the larvae wouldn't develop into the second star or third star stage and the larvae would die. So we wouldn't get a fly-strike lesion. That's what we are targeting, like not getting a fly-strike lesion on the sheep. So if we target the first instar larvae, which are most vulnerable compared to the second star and third instar, and that way we can control the pest without getting an advanced wound on the sheep.
Okay, so it sounds like you've got a bunch of candidate genes that you're going to look at, but it's not really clear what the mechanism will be to use those genes for pest control. Is that right?
So my point was to find a protein that induces lethal effect at the very first stage or the first instar stage, and to prevent the development of second star and the third instar, and thereby to get to prevent the formation of a fly-strike lesion.
Yeah, so I think, Simon, too, the other approach is so vaccination is one possibility, but the other possibility is a new class of insecticides that we might be able to come up with, if away from the current targets, which might then give us buy us more time with by having a new insecticide on the market to mix with the current bunch while we still work out whether it's feasible to do the vaccination.
Okay, thank you.
Is that right? All right. Are there any other questions? I'm conscious of time. Does anyone else have any other questions? All right. Well, look, if not, then we'll wind it up. If the Gothami's advisory panel can stay on the line, we'll have a bit of a discussion now about her seminar. And thank you, everyone, for your attendance and for your questions.